My name is Henk-Jan Hoving and I'm working in the research unit Evolutionary Ecology of Marine Fishes of the Department of Marine Ecology. My research area is the pelagic ecology, so ecology and biology of pelagic animals and deep sea animals. The open ocean is the largest habitat on the planet, and most of the ocean actually consists of deep sea. And of the deep sea, actually the largest part consists of the pelagic ocean, so the water column above the sea floor. And still, despite its importance in the global oceanic ecosystems and also in climate, very little is known about the communities in the pelagic ocean. Actually, whenever you take a new instrument into the ocean, it will result in new discoveries, and this fascinates me. In order to understand biodiversity laws, it's important to establish baselines. And this is basically the start from your time zero. And since the open ocean and deep sea ecosystems are so poorly explored, for many regions we don't have baselines. And that's exactly one of the things we want to find out with Pelagios, to establish a baseline of these faunal communities in the open ocean to understand the impact of ocean change. So one of the objectives of my research and the Pelagios, and why we're performing the Pelagios deployments, is to better understand high resolution depth distribution of animals in the water column. And we do so, we want to correlate these distributions and abundances in relation to oxygen levels. So in the Atlantic there's an oxygen minimum zone around 350, 400 meters. And what we want to know is how animals are distributed in relation to this oxygen minimum zone. And by studying animals, or by determining animal species that can live in these oxygen minimum zones, it will allow us to predict which are the winners and which are the losers of an expanding oxygen minimum zone in a future ocean. So the losers are basically the animals that can not cope well with low oxygen conditions, and these are, for example, fast swimming fishes, and it has been shown already in studies on marlins in the eastern tropical Atlantic that use a lot of oxygen, and their habitat has actually shrunken as a result of oxygen minimum zone expansion. Right now I'm on the Maria Esmerian on the journey number 61, where we have an interdisciplinary research group. And one of the things we want to find out during this cruise is how the ecosystem of Sangor Seamount, how it is built up, so what components does it consist of, but also how does it change throughout the year. So using this optical instrument into the deep sea, we've revealed a new biodiversity for this area. So some of the organisms we have seen were known to science, but they were not known to occur in this area. So this is an example piece of footage that we collected with Pelagios, the pelagic in situ observation system. And this is a squid that passes by the camera. Many animals that we see are actually gelatinous zooplankton, so large jellyfish like this Solmissus. So some of the challenges that you encounter when working in the deep sea is that first of all, you need technology that can able to withstand all these the high pressure of the environment. This results in specific technology. So you need specific housings, you need specific lights, you need specific electronics that can withstand these environmental conditions. Another challenge is that it's completely dark. So you need somehow to illuminate this environment in order to film it. So you need a lot of light, and lights take a lot of battery power or electricity. So you continuously challenge with how many sensors and how many lights can I use with the limited battery power that we have. So I want to learn more about these animals that are so hard to catch and so hard to observe in their natural habitat, and that fascinates me, and that drives me to develop new technologies and to go on research cruises into interesting ocean areas to research these organisms.